Hello, my name is Alex Johnson. I am a member of the QIAGEN Digital Insight Support Team. In this video, I will introduce how CLC tools can be configured to automatically analyze multiple samples individually using batch mode. Batch mode allows analyses to progress automatically, so as soon as one sample is finished, the next sample begins the analysis workflow.
Let's use the map reads to reference tool as an example. In this first example, each set of reads corresponds to a biological replicate. These reads are arranged in folders A, B, and C. Our goal is to map the reads and output a reads track for each sample to be used in the downstream analysis. First, I'll launch the map reads to reference tool. The batch mode checkbox is located at the bottom left corner of the input window. We can add the folders containing the reads by clicking the arrow. The batch overview defines the batch units, which in this case will be each input file. From here, I will click through the remainder of the wizard, leaving the options as default. The Processes tab lists which batch unit is running and how many samples are remaining. Notice how each file is processed independently and there is a corresponding output for each file. Once the work completes, we have 9 read mappings total, which is what we expect for this run. If we wanted to analyze a set of files at once, perhaps because we had technical replicates, we can uncheck batch. Next, I will select the reads and set up the map reads to reference tool again, but this time leaving the batch checkbox unchecked. I've already run this analysis previously, so we can just view the outputs. This will output a single result, which is comprised of all of the reads from each file. Notice the name of this file corresponds to the name of the first file that was added in the input window. The file can be renamed by right-clicking the file name in the navigation area and editing as needed. If we had a more complicated batching scheme, for instance, multiple samples with multiple technical replicates, it will be best to use metadata to designate which file goes with which sample. Please see our other videos to learn more about creating metadata tables and how to use them to configure batching. If you have any questions or concerns, feel free to reach out to the QIAGEN Digital Insight Support Team through email, chat, or phone. Our contact details are listed here, and thanks for watching this video.
QIAGEN.
Sample to insight.